All right, welcome to the pre-lab lecture for the LDH lab. In this lecture, we're going to talk about the techniques you'll be using in labs 3 and 4, weeks 3 and 4 of this lab. So just as an overview, in the first week, we started purifying LDH from our beef heart samples. We did this using a number of different purification techniques, including homogenization, salting out, centrifugation, and dialysis. Then last week during week 2, you finished purifying LDH using affinity chromatography, and then you prepared one sample for analysis using that desalting spin column. So for this week and next week, for the next couple of weeks, we are going to start characterizing our purified LDH. We'll do that using two assays, an activity assay and the Bradford assay. And then finally in week 5, we'll finish up characterizing our LDH by running gel electrophoresis to visualize the proteins that we have in our sample. So to get started with thinking about the techniques we're going to do for the next couple of weeks in lab, the measurement of several different types of volumes are going to be really critical. So to measure two different categories of volumes, we'll use two different types of pipettes. So for larger volumes that are greater than 1 ml, we'll be using graduated pipettes. And for smaller volumes that are less than 1 ml, we'll be using micropipettes, which are also sometimes called pipettement. So first, let's discuss the use of graduated pipettes. So graduated pipettes come in a number of different sizes. The largest ones we'll be using for this lab are the 10 ml size, and then they go down to 5, 2, and 1. So the way you read them is somewhat similar to the way you read those burettes during our first lab. And so just like with the burette, these graduated pipettes have the 0 line at the top, and then the numbers increase as you dispense liquid out the pipette. So for example, in this 10 ml pipette, if you wanted to measure 4 milliliters, you'd actually have to subtract that desired volume from the total volume of the pipette, which is 10. So if you wanted to dispense 4 milliliters, you would draw up liquid up to the 6 mark, and then you can dispense out that liquid fully from the tip. And so again, to arrive at that number, you have our total volume, 10 mils, and then you'll subtract out the reading, 6 mils, to give you the actual volume that you just measured and dispensed, which is 4 mils. So our 5 ml pipette works very similarly. You'll draw liquid up through the end, and then you'll have to subtract out to determine the actual volume that you are dispensing. So in this case, let's say you wanted to measure up to the 1.4 mark. If you then were to dispense that volume of liquid fully out, you would actually be dispensing 3.6 mils. Our 2 ml pipette is a little bit different. In this case, the bottom line reading here is actually the full 2 mils. So if you were to draw liquid up to the 0 here, and then dispense that liquid out only to here, only to this tube, that would give you the full 2 milliliters. If you actually continued to dispense out all the liquid, even from the tip area, you would actually be dispensing out more than 2 milliliters. So be particularly careful when you're using the 2 ml pipette. And the last one, the smallest one, is the 1 ml graduated pipette, but we, for the purposes of this lab, will actually be using micropipettes for volumes smaller than 1 ml, so you won't have to worry about them too much for these labs. So to actually draw liquid up and dispense liquid out of these graduated pipettes, we'll be using these thumb wheel dispensers for our pipettes. And so the different colors correspond to different sizes, and just like the name suggests, you use your thumb to roll the wheel. If you roll the wheel upwards, that will pull air or liquid up through the pipette tip, and if you roll the wheel downwards, you will dispense liquid out of the pipette. And so the different colors are able to draw up and measure out certain volumes of air, and they will correspond. In this lab, we'll be using only 2 of those 4 colors. We'll be using the green thumb wheel for our 10 milliliter and 5 milliliter pipettes. The green ones can go to a maximum of 10 milliliters, so they'd be able to also handle the 5 milliliter. You'd use the blue one when you're using your 2 milliliter graduated pipette. So just keep that in mind. Don't use a blue one if you want to measure a volume more than 2 mils, because you won't be able to. So let's then look at how we're going to measure out volumes smaller than 1 milliliter, and we're going to, again, do that using a micropipette. So very importantly, every time you use a micropipette, you're going to want to put a disposable tip on the end of the pipette. The disposable tip is the only place where the liquid you're measuring should go. You should never ever draw liquid further up into the shaft of the pipette. That can both be hurtful to you. If you get liquid up there, you can start cross-contaminating all of your samples with the liquid you got in there. You can also mess with the calibration or the actual measurements that are being measured or dispensed. So please be very, very careful to keep liquid always in the disposable tip. So when you're ready to use the micropipette, you have to be also concerned with some of the parts at the top. So to adjust the volume that you want to measure and dispense, you're going to use 2 different areas. The first one you'll use is the large volume adjustment knob. And so if you want to make a large change in the volume from what it's measuring now to what you want it to measure, start using the large volume adjustment knob. And then for more fine-tuned adjustments, you can use the thumb wheel, which is also called the fine volume adjustment ring. Once you've set your volume and you place your tip onto the shaft, which is also called the tip holder, and you measure out your liquid, then you'll need to eject your tip, because you'll want to use a new disposable tip every time you measure a new liquid. So to eject the tip, you'll press this tip ejector button with your thumb. That button will then push down this actual tip ejector, which will push the tip off of the shaft of the micropipette. Just be aware that sometimes the tip ejectors don't always work. Depending on the brands of the tips and the pipettes that you have, and the particular size of pipette tip that you have on the shaft, the tip ejector may or may not always work for you. So if you push the button and the tip doesn't come off, you can always just use your fingers to gently grasp the tip and pull it off the end of the pipette. So if you look at the front part of the pipette, that is where you'll actually see the display of the volume that you are measuring and dispensing. So it's very important that you are able to do that accurately. So that is what we're going to start talking about here. So first, when you want to measure out a particular volume of liquid, you'll need to select the correct size of micropipette and also put the correct tip onto the shaft of the pipette. So the different pipette sizes are often labeled in these different colors on that knob on the top, so that can make them easy to identify when you're in a hurry. But always double check by reading. There's always a label that tells you the pipette size, and it will often also indicate the volume range. So please be aware of these numbers so that you know that you're measuring the right liquid the right way, in the most accurate way possible without damaging any equipment. So let's start talking about the different pipette sizes. So the first one, the smallest one, is orange, and it's called a P2. The 2 in the name indicates the high end of the volume range, and often the volume range is tenfold. So if the maximum volume is 2 microliters, here we divide by 10 to get 0.2 microliters as the smallest end of the volume range. Our next size pipette going up is the P10, which is often red. And again, that 10 tells you the largest volume it can measure, 10 microliters, and that tenfold range, 1 to 10 microliters. Next, we have the P20, which is this pale sort of light yellow color. Again, the maximum volume is 20 microliters, and you have that tenfold range down to 2. The next size up is your P100. That one is often labeled in a sort of light pink or pale orange. Maximum volume 100 microliters. But here the range is actually a little bit smaller. It's only a 5-fold range. That goes down to 20. So be aware of that, which if you want to measure volumes in that range between 10 and 20, don't use the P100. The next size up we have is the P200, which is often labeled in yellow. Again, our maximum volume is 200. And here again, we don't quite have that tenfold range, but we can only measure down to 50 microliters using a P200. If you want to measure less than 50 microliters, you'll want to select a different size of pipette than the P200. So finally, our largest pipette size is the P1000, which is often blue. Measures up to 1,000 microliters, which, as we know, is equal to 1 milliliter. And also be aware that the range here isn't quite tenfold, it only goes down to 200 microliters. So once we've selected the correct size of pipette based on the volume that we'd like to measure, the next thing we had to do is select the size of that disposable tip that we put on the end of the pipette. So the smallest tips that we have, available are the 10 microliter tips. These are often clear in color, but the colors do depend again on the brand occasionally, so you can visualize to see what color the tips are, but always check on the box to tell you that will always tell you the volume capacity of the pipette tip. So a 10 microliter pipette tip, as the name suggests, can only hold up to 10 microliters of liquid. So you only want to use these smallest tips on our two smallest pipette sizes, the P2 and the P10, because those will only try to fit up to 10 microliters into that 10 microliter tip. If you want to measure larger than that, our next pipette tip size is the 200 microliter tip, which is often yellow. So you'll always want to use a 200 microliter tip when you're measuring using your P100 and your P200. Again, you don't want to try to ever measure more than 200 microliters into that 200 microliter tip, because you'll overload the tip. You might get liquid up in the shaft of the pipette, and then that will take a while to correct. So please always be careful when picking those tips. The largest tip size we have is the 1,000 microliter tip, or the 1 mil tip, and these you're always going to want to use on your P1000. So you'll notice I skipped one, which is the P20. So the tip size that you use for your P20 actually depends on the volume that you're measuring. So if you're measuring small volumes, 10 microliters or less, you'll want to use a 10 microliter tip. This will give you more accurate measurements when you're using the P20. If you're using larger volumes, between 10 and 20 microliters, you'll need to use a 200 microliter tip, because you never, ever want to try to put more than 10 microliters of liquid into a 10 microliter tip. So since this is a P20, your 1 to 10 microliters can go in the smaller tip, but your larger volumes will have to be measured in that larger tip. Alright, so now let's talk about how to actually measure out accurate volumes on a micro pipette. So depending on the pipette size that you're using, you will have displays that look slightly different. So let's start by looking at the P200. You'll notice that all the numerals here are in black, and the way we always read the numerals is from top to bottom. So if you look here at our minimum volume, you're reading 050 or 50 microliters, which is what we have here. And if you want to add a further significant digit to that, you can actually use these lines on the bottom and your fine volume adjustment knob. Excuse me. I just skipped too much slides. Okay. And your fine volume adjustment knob to actually measure even more accurately. So you could, if you wanted, in this case, we're reading exactly 50.0, but you could, for example, read 50.1 or 50.2. So reading the P200 is fairly straightforward. You can simply read the digits going down. You can add an extra decimal place using the lines on the bottom. So in this case, the maximum volume, you have 200, that's 200 microliters. You're exactly on the 0.0, so you're 200.0 microliters. Next, let's consider the reading from the P20 pipette. So here you'll notice that some of the numerals are black and some of them are red. And so here the red indicates a decimal point or a change in decimal place. So when we look at the minimum volume here, reading down the two black digits are 0, 2, so you have 2, and then red indicates decimal place, so 0.0. And then again, we can use these lines down here to give us one extra significant digit. And here this arrow is pointing right at the 0 line, so you have another 0. So in this case, the minimum volume that we're reading is 2.00 microliters. If we wanted to read a middle volume here in the intermediate part, we have our black digits tell us 12, and then the red tells us our decimal point is 5, And then since our arrow is pointing right onto this line in the middle, you have another 0. So you're reading 12.50 microliters. So lastly, we have the P1000 pipette. So here there's also a red digit, but instead of being at the end of the numbers as you go down, it's actually at the beginning. And this is also telling us a sort of change in decimal place. The way that I find most intuitive to think about it is to actually think of it as reading your volume in milliliters. So here, if we consider our maximum volume, you have our 1, and then the red to black change indicates a decimal place, and then we have 0, 0, and then again we can use these finer lines to give us more digits, so we can add one more 0. So this is 1.00 milliliters, which we know is equal to 1,000 microliters. The other way you can think about it, if it is more intuitive for you, is to always add a 0 onto these measurements. So here, for example, if we consider our intermediate reading, we have a 0 red one, and then 75, and then add on one more 0 for our decimal place, and that's 750 microliters. If you want, you could also read this as 0.75 milliliters, which we know is equal to 750 microliters. And again, don't forget, we can add on one more significant digit by looking down here, which in this case is another 0. But if it was moved a little bit to the left or right, it might give us something more like 752 microliters or 748 microliters if you were to just shift it over by one of those little line markings. So that is the basics of how to read and set the volumes on your micropipettes using the 3 different scales that we see here. So now that we're all ready, we put the correct tip on the correct size of pipette, and we set the volume that we want to measure, which is within the range for our pipette. Now, how do we actually use that pipette? So this diagram shows you the number of different steps you'll have to follow. So the thing you want to be really careful about here is our plunger button, the white knob that you can turn to set the volume. So it has essentially 3 different settings where it can be. It can be fully out or fully up, which is shown here on the blue line. That's how it is most of the time, unless someone is pushing on it. And then when you want to start drawing up and ejecting liquid, that's when you'll have to start pushing the plunger down. And there are 2 different places where the button will stop. What's shown here on the green line is what's called the first stop. The location of that first stop will vary depending on the exact volume setting that you have set on the micropipette. If you're only going to be measuring out a smaller volume, that first stop will be higher up. You'll move the button less. If you're measuring a larger volume, you'll usually that first stop will move down, and so you'll end up pushing it down a little bit more until you feel that first stop. There's also a place shown in red here that we call the second stop. The second stop is where you get, if you push the plunger button fully down, you'll feel a place at the first stop where you'll feel some pressure back from the button trying to stop, and then if you keep pushing past that, you'll get to the second stop where you cannot push the plunger button down anymore. And we want to be very, very careful about when we're using our second stop because that can potentially damage the pipette if we do it at the wrong time or the wrong location. And the same thing with the first stop. So we want to be really careful about knowing the difference between those different stops. So keep that in mind as we go through how to use the micropipette. So we start out, the plunger button is fully up, and we've loaded our tip, we've set our volume, and we're ready to go. So before you put that tip into the liquid you're trying to draw up, you'll depress the plunger button down to the first stop, to that green line. And you want to do that when the end of the tip is in the air. If you do this when the end of the tip is in a liquid, you will blow air bubbles into that liquid. And that can make a mess and also make it harder for you to draw up that liquid accurately. So please be careful to depress to the first stop before that tip even goes into the liquid you're trying to draw up. Once you press the plunger button down, now you can place the tip inside of the liquid you're trying to draw up. And then to draw up the liquid, you raise up the plunger back up to that top position. Once you've pulled the plunger up to that top position slowly, you can then pull that tip out of the liquid that you're measuring. At that point, you have put liquid inside your pipette tip. Be really, really careful not to, when you're holding your pipette with your hand, don't at this point tilt the pipette so that it's horizontal or even upside down. If you do this, the liquid that's in the pipette tip will follow gravity and it could go up into the shaft and into the micropipette. And again, we'll have to take the whole pipette apart and clean it and potentially mess with the calibration. So please make sure to keep the pipette in this upright position, especially when you have liquid in the tip. So that being said, now we want to release that liquid into a new container. So once we put the tip under the tip into the new container, you can start depressing the plunger down to the first stop, and that will release the liquid that you measured. Sometimes, after you've pushed down to the first stop, there will still be a little teeny tiny bit of liquid right in the end of the tip. And if that's the case, this is that one time when you can use the second stop, and then you can expel that little tiny drop of liquid that's stuck on the tip. And even once you've pushed that plunger fully down, you'll sometimes need to actually touch the drop to the wall of the container to kind of encourage the drop to come off the end of the disposable tip. So once you're fully dispensed at this point and you're ready to pull the pipette out, don't let go of the plunger until the tip is out of the tube. Again, when you let go of that plunger, it's going to pull up, and it's either going to pull air or liquid up into the pipette. And especially when you're at the second stop, when you're at the second stop, you only want to pull air back in with you. You don't want to accidentally pull in liquid from this tube you just filled. So only let go of the plunger when your liquid is fully dispensed and your tip is out in the air again. I also just want to add that if you're new to operating a micropipette, I highly encourage you to practice drawing up and dispensing water, setting volumes, all that good stuff. Try it on water before you use any of your samples or reagents. That way you'll feel more comfortable when it comes time to do the real thing.